Yes, good evening to all the participants. Welcome to the day 7 session on molecular phylogeny. Today, we'll be discussing on phylogenetics basics and how to work on phylogenetic trees using phylogenetic tools. And then we also discuss about the methods for construction of phylogenetic trees. Before going to this session, can we go ahead with the recapping of the last session contents? Yeah. So what are the last session topics? So good evening. So you can, yes, very good. So we have discussed on blast and FASTA. We discussed on E value, bit score. You remember where, the E value, bit score, whatever formulas I have showed is for blast tool. Okay. And we also have types of blast. Majorly we discussed on 5 versions of blast. Like, can I ask you the second question? What are the major 5 versions of blast? Very good. So I got from Bhagya Lakshmi, that is blast 10, blast P, blast X, T blast, and T blast. Is it so? This is right? No. One thing is wrong here. What is that? Can you identify that? So out of these 5 blast, one blast is wrong. What is that? We have blast 10, that is nucleotide query versus nucleotide. This is protein versus protein. This is translated nucleic acid versus translated nucleic acid database. And this is translated nucleotide versus protein database. Whether we have T blast, so you're not confident. Actually, this is one of the question asked in TAT, isn't it? So what is then? Instead of T blast, we have discussed what? Let me show you the lecture. Yeah. So what is that? Instead of T blast, we should have T blast N. Okay. Any participant has answered? No, I think you have to go through because like this only you can find questions in competitive examinations. Okay. Yeah, and we also discussed on formulas of E-value and bit score for blast. We have discussed on basically like expansion of PHI and PSI blast. Can someone expand PHI and PSI blast? Ex- yes, you can also unmute and answer. What is
Associated block
ex- exactly? And PSI method?
specific iterated block.
You are right, madam. Actually, like here in position specific, we bother on the amino acid positions if it is a protein. And if it is a pattern specific, generally we go for exon specific for a nucleotide, or we go for the motif specific for a protein. This is how we check the patterns. Very good. So nice try from your side. So we have discussed also the algorithms, blast and faster in overall, isn't it? So can someone tell me how many steps are there in blast algorithm and how many steps are there in FASTA algorithm?
So 3 steps in blast and 5 steps in FASTA.
Exactly, madam. 3 and 5. So actually, here the fifth step in FASTA is to calculate the E value. Okay. So this is how we have discussed on the blast and FASTA algorithm. Now we can go ahead on today's session on molecular phylogeny. So in molecular phylogeny, okay, the phylogenetics, this is an application of multiple sequence alignment. That is, if and if you have pairwise sequence alignment. The output of pairwise sequence alignment, we can give that as an input for multiple sequence alignment. And whatever output of multiple sequence alignment, we can feed that as an input for phylogenetic tree construction. So if so, yes, a participant can unmute. Okay, now let me continue on the phylogenetics. In phylogenetics, okay, we are taking an input of multiple sequence alignment, or else even we can take multiple sequences as an input. Okay, so we have different types of raw inputs for for construction of phylogenetic trees. Then what is phylogeny actually? So phylogeny trees illustrate the evolutionary relationship among groups of organisms or among a family of related nucleic acid or protein sequences. So for example, if we take a tree here we have mammals or close resembling to turtles, but not related to snakes and birds if you compare turtles. And if you see the turtles are closely related with mammals and snakes. And of course, we have a large tree, I mean, if you see this branch, okay, the branch, this length to this length is small when compared to this length and this length. So, turtles are closely related to snakes when compared to birds. Okay, so even based on the distance length, one can able to determine what species are closely related. So this is a phylogenetic tree, and we can able to see something like a common root through the it's branching from a common root, and the inference of relation we can make based on the close proximity. The mammals are is closely uh, linked with turtles just because it's in close proximity, whereas the snakes and birds are having a distant relation. Now, if you see what are the website URL for practicing phylogenetic trees, so we can see interests. Okay, in interest, you can have a taxonomy. One can even find the taxonomy to find the kingdom of vertebrates. So, what are the group of classification of organisms and their vertebrate? And if you want to work on ribosomal database project, so one can go with this URL. And for tree of life, we can go with this URL and file it. This is a most commonly used tool for construction of phylogenetic trees. That is PhyLib. PhyLib means phylogenetic inference package. So in this phylogeny inference package, almost we have 35 programs to work on construction of phylogenetic trees. So this is one such widely used tool for this is a package widely used for construction of various phylogenetic trees. And if you want to go for specific methods, we have different tools. One such tool is PAUP. So before going to this, we can discuss about what are the types of trees and what are the methods, different methods for construction of phylogenetic trees. Now let me go to the next slide. So in this slide, we can see the types of phylogenetic trees. Generally, the trees are classified as rooted tree and unrooted tree. So how I can able to tell it is a rooted tree? So if you find a common root, if you find a common root like this, and from this you can find A and B are branched and C and D are branched. Okay, A and B are sub branches of this main branch, and C and D are sub branches of this main branch, and E you can find it's a like a main branch. So according to the terminologies, the parts of trees, we can classify each and every uh, character mentioned here, or what we call it as nodes. Okay, this we call it as node. Okay, what do you mean by node here? Like here you have five different sequences. For example, if you are comparing insulin, okay, of five different species, can be the A is considered as human, and B is considered as pig, Okay, and that is, and if you so scrofa, and if you have chimpanzee and not chimpanzee gorilla, because we already seen the insulin ligand is the human is more closely related to gorilla. So in our example, whatever we worked, we got human and gorilla are linked with each other in insulin ligand protein, and chimpanzee, that is pan troglodytes. Okay, and the bonobo macaque monkey and bonobo chimps also uh, resemble similar. That is, chimp and macaque monkeys, the gibbon monkeys, have a close proximities. And you can find other thing like Bos taurus. Bos taurus is something like is an ox. Okay, so they're having a distant relationship. Okay, so if you have a common root, and if the common root, if you find so many branches, we call this as rooted tree. But here in this unrooted tree, we don't have any branches. I mean, we don't have a common branch, but we have multiple branches. Where can I have a common branch? I can't, because we can't able to have a common root. So we call this as unrooted tree. Okay, and here the parts of the tree are nodes, and here you have types of nodes like external node, whatever you see at the final. Okay, so I take a different color for representing. So you whatever you see here, okay, this one is an external node, this is another external node. Okay, so the nodes obviously represent the sequences of a protein or a nucleotide. If this is an external node, okay, we also have a different uh, node which is present inside. This is what we call it as internal node. The internal nodes are present inside, the external nodes are present outside. Obviously, the external nodes present the sequences. The internal nodes are not like that, it's a branching. Here, if you see, this is the internal node for the rooted tree. This is also internal node for the rooted tree. Of course, this is also an internal node. Okay, but if you see the external nodes, okay, the external nodes of this rooted tree, you can find here. So these are the external nodes. External node 1, 2, 3, 4, 5. 5 external nodes you can able to find this rooted tree. So you can able to find in this slide about the types of phylogenetic tree. One is for rooted tree, the other is unrooted tree, and these are the parts. Okay, so nodes. You have the types of nodes like internal node and external nodes. Okay, in both the types of trees. Now let me go to the next slide. So in this slide we discuss on molecular clock theory. So according to the molecular clock theory, we can have three kind of hypotheses. The first hypothesis, the initial hypothesis was proposed by Zuckerland and Pauling in early 1960s. So according to him, for any given protein, accepted mutations in the amino acid sequence for the protein occur at constant rate. Okay, for example, if you take the human eye, okay, or not human eye, you can take anything else, because even in eye you can find major difference between chimpanzee and human. But if you take protein like insulin, okay, even in insulin, when compared to chimpanzee, you can find gorilla amino acid sequence of insulin is having higher similarities. But I told you, if you take any other proteins like hemoglobin, myoglobin, so these proteins like transport proteins and muscle proteins resemble more closely resembling to chimpanzee. So what do you infer from that? So you can infer that if mutations are happened, even in case of insulin protein, it occur at constant rate. Okay, so that is you have a small variation with respect to each and every ancestral species. Okay, but it won't apply for all the proteins. For most of the proteins, the evolutionary rate, that is the mutation rate, is consistent, is at a constant rate. Okay, so for example, if you compare human with so humans are coming under mammals. So take an example like mammals. After before mammals, we have the birds, isn't it? And before birds, we have reptiles, isn't it? So if you have these kind, okay, so if you whether you can able to compare ostrich and human, you find wide variations with respect to phenotype. Okay, isn't it? So each and everything, the ostrich can't be compared with humans as such. So we find variation with respect to limbs, we find with respect to eye, with respect to the nose, ears, etc. Okay, so a lot of changes will be there. But if you take closely ancestral species, we have most of the phenotype, except it has a kind of a constant mutation rate. So that's what the Chackerland and Porling in the early 1960s, they have proposed this hypothesis. The second hypothesis, that is according to molecular clock theory too, the rate of accepted mutations may be different for different proteins. Okay, may be different for different proteins depending on the toleration for mutations. So that's what. So even in insulin, you can see instead of chimpanzee, we got gorilla as the most closely resembling. So, and even if you take few proteins, I told you an example like hemoglobin, myoglobin, we find human and chimpanzee share the common amino acid characters in the protein sequence. If so, okay, we can able to find that the evolution rate, okay, the mutations in each and every proteins, okay, are not dependent on all the proteins, but it varies with protein to protein. And so different parts of protein may evolve at different rates. Okay, so this is molecular clock theory 2. And according to the molecular clock theory 3, if A and B differ by K accepted mutations, then roughly K by 2 mutations have occurred since divergence. The K by 2 mutations are proportional to time since divergence by clock theory, and so full matrix of data can be collected. Okay, so this is something related to the last molecular clock theory. We have K by 2 mutations. Actually, if you see the evolutionary rate between human and chimpanzee, okay, human and chimpanzee, so think like, so this if you consider as a chimpanzee, okay, human to chimpanzee, okay, so you can able to find around, okay, the evolutionary rate between if this is chimp and if this is human, okay. So the evolutionary rate between the chimp and human is around 10,000 million years. 10,000 million years. Okay. According to the according to the information, okay, gathered. So like if you see in bioinformatics textbooks, they might have mentioned this span to evolve from one species to the another species. And if you take a gorilla and chimpanzee, again, okay, if you take a gorilla, okay, again, you can find a span of 10,000 million years. So for each and every species, okay, so in bioinformatics textbook, they have mentioned that for each and every species evolution, it has taken a time, a consistent time of 10,000 million years. Okay. So if you have this, okay, if you find an evaluation, okay, a change in evaluation can also occur by K by 2 mutations, okay, according to the clock theory 3. Okay, now let me come to the a large phylogenetic tree. If we have multiple species, more than 100 species, then how to find the tree? The tree, instead of finding a normal rooted tree, okay, you can have in the form of this kind of a tree. This we call it as a radial tree. Okay, so if you have a multiple species, one can find this. This looks like a pleasant experience. Instead of showing like this, okay, instead of showing like this rooted tree, one can go with this kind of a tree where you can find almost all these characters significantly can find this. Okay, and now before stepping into the yes, we can first step into this phylogeny flow chart. That is how to what are the different methods of phylogenetic tree construction and how to select a method for construction of phylogenetic trees. For this, first we discuss on methods of phylogenetic tree construction. So if you see the methods of phylogenetic tree construction, the first method what we see here is character-based method. This is a character-based method is the first method of phylogenetic tree construction. And the second method is the distance-based method. If you see the character-based method, okay, we have only two methods. One is maximum parsimony and the other is maximum likelihood. And if you see the distance-based methods, we have plenty of methods for phylogenetic tree construction. Number one is UPGMA. UPGMA means unweighted pair group method of averages. So this is the expansion of UPGMA. Okay. And if you see NJ stands for neighbor joining. And we also have another method called weighted neighbor joining method. FM stands for Fitch-Margolish and ME stands for minimum evaluation. So these are the distance-based methods. Okay. And if you see this distance-based methods, okay, we also have least squares. Okay, this is an additional method that we are showing here. And these whatever methods I classified as a character-based or distance-based, okay, this is extracted from a textbook called Bioinformatics by Mount. So in universally throughout the globe or in most of the countries, if you see the basic textbook for bioinformatics, we follow this Mount. Okay, one can go through this information from this Mount textbook. Okay. Now, if you see the second thing, okay, other than classifying the methods like character-based or distance-based for construction of phylogenetic trees, so we can categorize these methods, okay, also based on clustering and objective function. So if you're using a clustering, okay, so there is no such character-based methods present. So you don't find any character-based methods present under clustering. But we have on the objective function based, that is maximum parsimony and maximum likelihood. And if you see in distance based, we have UPGMA and neighbor joining. And if you see in under objective function, we have minimum evaluation and least squares. Under objective function, that is using the functions, one can able to construct a phylogenetic tree using minimum evaluation and the least squares. I think you already come to know about these least squares in numerical methods, isn't it? They are using we use a functions for calculations. But here it's not like that in this UPGMA neighbor joining, where you cluster the data. Okay, we cluster the data for construction of trees by stepwise method. Okay, so that is not followed in character-based method. So to know more about this clustering objective function and even more about the characters and distance-based methods, we can discuss in subsequent slides. Okay, so now we have completed how to categorize, what are the different methods of phylogenetic tree construction and how to characterize these methods based on clustering and objective function. Now we discuss on phylogeny flowchart. So if you see the methods, we have plenty of methods, isn't it? Two methods here and four methods I've shown there. And earlier in another slide, I've shown two more methods. So totally six methods we have. Okay, we have more than six methods also. So we have plenty of methods. Then how to select a method for phylogenetic tree construction if you have a family of sequences? You remember on yesterday, I was taking insulin ligand protein and just compared with four more species. Okay, so if I have family of five insulin ligand protein, okay, how to build an alignment? So how to construct the trees? So to construct the trees, the first step what we make is build the alignment. Here, whatever alignment we build is multiple sequence alignment, MSA. First, we have to construct a MSA, and after that, based on the similarities, so how we can identify the similarities? The similarities based on the number of matches. If you have more number of matches, more than 95% or something like that, okay, better we can step on to use the character-based methods like maximum parsimony or maximum likelihood. So don't follow this. Okay, so you can't. You just ignore this. Okay, you just ignore this. So either use the maximum parsimony or maximum likelihood methods. And if you have recognized similarity, so that is this can take like above 90 or 95 percentage. But if you have less than 90 percentage based on your project, you can consider this as a recognizable similarity, and one can go to the distance-based methods. So in some literature, I mean, some journal papers they have taken 95, and some journal papers they have taken 95. It's your choice then. If it is above 90, okay, the minimum thing you can go for character based. If it is less than 90% similarity, I mean the similarity of all the five sequences, whatever you take for comparison. In our case, for an example, okay. So this is what in distance-based methods you can choose based on the clustering and objective function. If you want to use a clustering based, you can use UPGMA or neighbor joining. If you want to go for objective, one can go for minimum evaluation and least squares. Okay. So this flowchart gives you how to select a method for phylogenetic tree construction. Yes. Now, in the next slide, we can see the formula to calculate the number of rooted trees and number of unrooted trees. Okay, so if you see the number of rooted trees, this is calculated from 2n minus 3 factorial divided by 2n minus 2 into n minus 2 factorial. And the unrooted trees are calculated through the formula 2n minus 5 factorial slash 2n minus 3 into n minus 3 factorial. For example, if you have 5 number of total sequences, they are using this calculation and the value is calculated. Okay, now I give you a simple task. If I take 4 number of sequences, okay, how many rooted trees you can get? So one can try this. I can half for you unless you take a pen and paper and you can try this. And how about for unrooted trees? See the formula, see the bottom calculation for your reference. Like 7 factorial, how come we can write 7 into 6 into 5 into 4 into 1 into 2 into 3, isn't it? So exactly how come we can write this 7 into 6 into 5 into 4 into 1 into, sorry, 3 into 2 into 1 divided by 2 power 3. 2 power 3 is nothing but 8, okay, and 3 factorial. In 3 factorial, 3 into 2 into 1, okay. So 3 into 2 into 1, 3 into 2 into 1 getting cancelled. So here, if you want to strike out a in the denominator, so here you will be getting like 3 and 4 will be stacked off. So 7 into 3 into 5. Okay, 21 into 5 is nothing but 105. Okay, so likewise, you can try with 4 rooted trees and for 4 unrooted trees. So if you got the answer, unmute and answer. Participants, presently I don't have power. Okay, even I don't think if I have any internet frequency disruptions, kindly let me know. Okay, am I audible to everyone at present? Yes or no?
Yes, sir, audible.
Yes, sir.
Yes, sir.
Yes, sir, you are right.
Good. Can you try this?
Sir, 105, sir.
For router trees, for 4 sequences, is it? No? You want?
No, sir. No, sir. Okay.
Yeah, you try this.
15, sir.
Exactly. Your answer is right. And for unrouted trees, can I try unrouted? 2 into 4, 8 minus 5, 3 factorial divided by 2 power 1. And here you have 1 factorial. It's very simple, isn't it?
3,
3
sir.
into 2. What is the answer, madam?
3.
Exactly, ma'am. So the correct answer is 3 for unrooted and 15 for rooted trees. Can we say this? Yes. For the 5, we have 15 and 105. And for 4, first we have calculated rooted, isn't it? 15. And for unrooted, we have 3. So likewise, if you try with the calculations, you can get this much number of rooted and unrooted trees. For the 10 sequences, see how many rooted trees and how many unrooted trees. So basically, one should have a good computational power, okay, to work on this tool if you work with multiple sequences. Earlier, I showed you more than 100 sequences, which I represented in radial tree. So for 100 sequences, see the calculation, how many rooted and unrooted trees you'll be getting. Okay. So of course, here for phylogenetic trees, you should need more computational power. Okay. Now we have discussed all about these methods and how to go for calculation of rooted and unrooted trees. Okay. Power is very. Okay. Now let me go for the practical illustration, okay, of how to work with um, phylogenetic trees. So for this, initially I'm going to provide you demo with Mega software. Mega. So Mega 7 I have installed. So this you can go to the major website URL. Okay, for going to the Mega software, I can also show you the URL. Yes, this is the URL, megasoftware.net. So for drawing the phylogenetic trees, one can go with a grape tree, ITOL tool. You know, this ITOL tool, one day I've taken the radial tree. So if you are having more than 100 sequences, more than 20 sequences, better go with ITOL tool for getting a phylogenetic tree in radial. And we have Mega software, MAFT file. Okay. Okay. Now let me teach you from Mega software how to work on phylogenetic tree using Mega software. File, first you have to open a file. Okay, the font can't be increased. I could just simply dictate file open. Okay, and I'm going to open a insulin file. So yesterday we have saved that insulin file. Yes, we have insulin 5 proteins, isn't it? So this I'm going to open. So when I'm opening, it is asking how do you like to open this FASTA file. So if I want to analyze, I can click analyze, or if I want to go for alignment, you can click align. It's a show. Presently, I'm giving like analyze, and it's asking whether it's a protein sequence or nucleotide. It is a protein sequence, isn't it? So now I click OK. Oh, there is error. I better go with file, open a file, analyze. Okay. Align. So better click align instead of analyze. After aligning one day, we can analyze. So now I clicked on align. So we got this window. Okay. In this window, one can able to see here Homo sapiens here, and I can find the 5 different species. So according to these 5 sequences, the Homo sapiens is closely related to Suscrofa, isn't it? We don't have gorilla, chimpanzee. So we have to get a result like that. Now we can check whether we're getting or not. For this, first I select all these 5 sequences. And if you want to go for alignment by cluster W, you can click this. If you want to align by muscle, you can click this. Okay, we have a types of MSA. For example, if I want to go with muscle, I click muscle. Okay, and I have to simply click compute. So after doing a multiple sequence alignment, okay, if I want to construct a phylogenetic tree, okay, so I can also able to create how to create it. So for this, so I already created an alignment. Okay. Now I'm going to work on tree construction. For this, click phylogeny, construct test. Mhm. Compute pairwise distances. It's asking mega file. Data phylogenetic analysis. Yes, I click data. Here I have phylogenetic analysis. I just click the phylogenetic analysis. Okay. After clicking phylogenetic analysis, you get this two information. One is TA and the closed data and this. If you want to see the alignment, you have to click this. If you want to see this TA, if you click this, you can also find a sequence data explorer. Okay, now presently we don't want the sequence data explorer. After building alignment, I'm clicking phylogenetic analysis. And after clicking phylogenetic analysis, after visualizing these icons, one can click phylogeny and construct tree based on any of the distance-based methods. If I want to construct based on the UPGMA method, I can click this. So it's asking, would you like to use the currently active data? Yes, I want to use the current data. Only after clicking phylogenetic analysis, I can able to find a mega file is created. A mega file, what we created is phyloanalysis.mega. So, and it's asking to use the currently active data. Yes, the present 5 insulin proteins I want to use. So that mega data I'm going to use. So I clicked OK. And now clicking compute for constructing a phylogenetic tree using UPGMA. So this is the tree we got for from UPGMA. Okay, and you can also find the distance scale. So according to this, we can able to infer the human is closely related to Suscrofa, Homo sapiens Suscrofa, and Homo sapiens and Suscrofa are closely related to Bos taurus, then Cavioporcellus and Dania rederio. So these are the relationship we can able to find from phylogenetic tree using Mega software. And if we want to find, okay, in different kind of trees, okay, like if you want to get in traditional, if you want to, this is a rectangular, isn't it? Instead of a rectangular vertical tree, this is vertical, not this is a horizontal tree. This is a rectangle. Uh, this is actually vertical. Is this okay? Horizontal. No, this is what we call horizontal or vertical. If this is horizontal, the vertical is we get in the form of columns, isn't it? So whatever tree we have, okay, if I want to have in different views, okay, for example, if I want to have a view like traditional inclined, this is an inclined tree. Earlier we had a rectangular tree, now we had an inclined tree. And similarly, if you want to increase the font, one can increase the font from here. And if you want to go to a specific node or branch, you can click here. Instead of an inclined tree, okay, if you want to have a curved tree, if you click this, this is what you can find. So if you find a tree like this, we call this as curvogram. Okay, we have a types of tree like I can explain you. Yeah. So we have types of trees. Okay. So if you have a tree like this, okay. Okay. We can't take this. Okay. So this is a rectangular tree. And I showed you something like this. I mean, so this is a curvogram. We also have soupogram. So different outputs, what we get is general defaultly what we call it as a phylogram. The output of tree, what we call it as a phylogram. The phylogram can be classified as phenogram and cladogram. So phenogram and cladogram. So if you're constructing a tree only with characters, considering only the characters, we call that as cladistics approach. If you're using a cladistics approach, and the type of output what we get is a cladogram. And if you're using a phonetics approach, phonetics here I mean using both characters and distance. Characters and distance. What is the character I mean? The character I mean, if you take a protein, amino acid characters. If you take a nucleotide sequence, the nucleic acid bases characters. So here you're using only the characters in consideration for construction of trees. But here use characters. Other than characters, I mentioned you as a distance. The distance, what I mean here is the genetic distance. Okay, so if you use both the genetic distance and characters, you get a phenogram. Okay, so these are the outputs of phylogram, and the subtypes of phylogram is mentioned here. So other than this, the types of output we can tell, like I told you just now, curvogram, soupogram. Okay, so these are some of the outputs we can obviously anticipate while construction of trees using different softwares. Okay, so this is something about the outputs. Now let me continue on the presentation. Yeah. So I was explaining you about that. Here you have something called time tree. What is this time tree? To know about the time tree, one should know what is the basic difference between a phylogenetic tree and evolutionary tree. Okay. So the phylogenetic tree and evolutionary tree, the terms looks very similar. But what is the major difference between the phylogenetic and evolutionary? So in phylogenetic trees, just now I was explaining, either it can construct, the output is phylogram, and it can either construct phenogram or cladogram based on what characters on distance, whether you're using characters and distance for construction of trees. Okay, but what is something called evolutionary trees? So in evolutionary trees, other than considering characters, distance, that is genetic distance, one can also use time. Okay, if you consider other than characters and distance, so it means that in a distance tree you can find a distance value. For example, you have a distance like 4, 3, 2, and 1.5, something like that. So in distance you have a distance value also, other than the characters usually used to construct the relationship between the species in the tree. Okay, so and if you see this time based, so other than the distances whatever we have mentioned, here you also have a time based. You remember I was explaining about the molecular clock theory, a hypothesis. For each and every species you can find an evolution of nearly 10,000 million years span. Okay, so if such a distance is there, okay, such a time gap is there, one can able to mention here. For example, mammals to turtles, you can mention something like 40 million years. And for turtles to snakes, so we can mention like 30 million. So based on the data available. Okay. So other than the distance values in the trees, one can also see their time scale. Okay, so this is an additional thing you can find in evolutionary trees. Okay, you got it. So This is something we can find in the tree. So this is a distance scale. This is the distance scale. So for example, here to here is point 1, but here to here is point 2, here to here is point 3. But if you see, if you want to even go for time tree scale, one can click here and you can also find the time. In addition to that, you can find this information. So other than this, if you want to swap, go for bootstrapping, there is a concept called bootstrapping. We can able to do using this mega software, flipping, etc. And you can also save this file however we want. And if we want to go for distance table, because the distance table can be created. Okay, distance table can be created by clicking distance and compute pairwise distances. Would you like to use currently active data? Yes. So now it's giving a kind of a window. Simply click compute. So you can able to see. So for the species, whatever we compared, that is five species, you can able to see in the data are present in the lower triangular matrix. It means that in the diagonal, if you fill the elements, like you have 5 into 5 matrix, 5 cross 5 matrix, in the 5 cross 5 matrix, okay, below the diagonal, you can find the elements. So we call this as lower triangular matrix. Then what are the elements you can find in the diagonal? One can able to find the zeros in the matrix. So how this distance value is calculated? So for this, I'm going to give an example. So I'm going to give an example how the distances are calculated through UPGMA. For example, I can give you here. So consider I'm going to use 5 sequences or 4 sequences. Sequence A, okay, is A, G, C, T, and the B sequence is A, G, C, C, and C sequence is A, C, 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 and D sequence can be created like C, 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 C. Think you have 4 different species. The first is human, the other is chimpanzee, the C is gorilla, D is for orangutan. Think like we have 4 species, and the 4 species particular DNA sequence, okay, is like this. So I'm just showing you with few characters. Now, if I ask you to construct a distance table, okay, one has to uh, create a kind of a matrix. So like A, B, C, D, I'm just creating a matrix in down A, B, C, D. Okay, I'm going to have as such a kind of a matrix now with the 4 sequences. Whatever 4 sequences I have, okay, I've constructed a kind of a matrix at present. Now, so according to the theory, okay, so initially if you are going to fill the lower triangular matrix, so then you have to fill the diagonal as 0, okay. After filling all these elements as 0, so you have to check, I have to fill these values 1, 2, 3, 4, 5, 6. To fill these values, first we can compare A and B. If A and B are compared, how many mismatches are there? So if A and B are compared, how many mismatches? You have only one mismatch. T and C alone is a mismatch, isn't it? So according to the uh, genetic distance, that is here in this case is a Hamming distance. Okay, the distance found is 1. So that you have to fill here. 1 here you have to fill. And if you compare A and C, okay, if you compare A sequence and C sequence, okay, you have 2 mismatches. One is this and the other is this. If you compare A and C. So one has to fill here as 2. You got it? Whatever Hamming distance you get, you have to fill it up here. Now B and C. B and C generally only one mismatch. And A and D, we have 3 mismatches. And B and D, 2 mismatches. C and D, only one. Isn't it? This is how we have to create a distance table if you are provided with the sequences. That in the distance table is nothing but genetic distance. For how many characters are varying on comparison. We are not bothered about matches. We bother about only mismatches and gaps on comparison of multiple sequences. Got it? So on tomorrow I'll be explaining you on how to construct a phylogenetic tree using UPGMA, a distance-based method. That is a problem-based, okay, a matrix-based calculation. Okay. For this is an essential step to calculate a distance table if you are provided with the sequence. So in the problem, either they provide the sequences or they provide this kind of a table, which is a derived output of the sequences. So from this, you have to take to construct such a kind of a tree. If you see this tree, okay, so that is one is closely linked with two, isn't it? Okay, so what? So obviously can find the same in Venn diagram. If you have two sequences, 1 and 2, okay, this is closely resembling. So when you get a kind of a tree like this, okay, and if you see 3 and 5, this also closely resembling each other. So it you have an Venn diagram like this, okay. And if you see the 4 is linked with 3 and 5, but not linked with 1 and 2. It's having a distance, okay, a distance apart. So you can have 3 and 5 in a circle and 4 in an outer circle. And of course, 4, 3, 5 are present in a circle. And of course, everything you are enclosing this circle. Okay, so this is how whatever you represent in the Venn diagram, we can able to create a tree, the same kind of representation, but in the form of a tree representation. So the relationships are clustered here. Okay, here we have clustered initially 3 and 5 as a single OTU. You know, there is a terminology called OTU. OTU means operational taxonomic unit. If you have DNA sequences in comparison, if you are comparing two different sequences, okay, here you are grouping. Initially, you have 5 OTUs because we have 5 sequences in this example. Okay, but while grouping, according to the step, one of the step in UPGME, you have to group OTUs. So here 3 and 5 are grouped as a single OTU, and whatever single OTU you have, you are grouping with 4, and now you are getting this entire 3 sequences as a single OTU. Already you have group 1 and 2 as a single OTU, and now this OTU, you are combining it as a overall OTU. Okay, operational taxonomic unit. So all these things we can able to observe, and these are one of the steps, of course, we can able to find in UPGMA, that is pairing. So about more about how to solve a problem using UPGMA, we discuss it on tomorrow. Okay, so this is something related to
Excuse me, sir.
tell me.
Sir, can you show me the slide of the rooted and unrooted tree, wala, sir?
Yeah. So I've taken to the rooted and unrooted. Now you can ask me
Sir, this one not formula, sir.
formula. Yes. You want?
Sir, can we explain the this rooted tree 7 factorial?
Good. Okay. For 7 factorial, you see, so rooted 3. Okay. 2 into
Yes, sir.
7 is 14 minus 3 is 11 factorial. Okay. Divided by 2 into 7 minus 2. That is 2 power 5 into 7 minus 2 is 5 factorial. Isn't it? 11 factorial. Here you can have something like 11 into 10 into 9 into 8 into 7 into 6 into 5 factorial. Okay, and here you have 2 power 5. 2 power 4 is 16, and so 32. 2 power 5 is 32, and 5 factorial you have, isn't it? For simpler classification,
Yes,
so,
yes, sir.
so a 5 factorial, now 32 is nothing but 8 into 4 is 32, isn't
32.
it?
Yes, sir.
So we have something like if you cancel this, you have 4 here.
4.
Yeah, so 2,
Now 2, 3.
3, and 2, you have 5.
5,
So totally 55 in
55, 5
9
to
into
9.
1. Okay, so this is
Yes,
the
sir.
calculation. So one can try this. So I use a calculator at present for this. So I'm going to use my calculator. So I'm
I can
getting like,
understand, sir.
oh, you got this now, no, isn't it? So the
Yeah, yeah.
exact answer,
Okay.
uh,
Thank you, sir.
you find you find the exact answer.
Yeah, yeah,
Uh,
sure.
10,395 is the answer. 10,395.
Yeah.
Okay.
Yes, sir.
So we can see this in the next slide. The sound 10,395. Okay. Yes. Good. Okay. So now we are going to see another tool for phylogenetic tree construction. So about solving the problem in EPGMA, we can see it on tomorrow. So if you want to work on, if you want to construct such a kind of a rooted trees, you know, so whatever rooted trees we have, okay, if you want to construct only the rooted trees in Phylip program, in Phylip program, we have, I mean, in Phylip package, you have a program called drawgram. In a Phylip, you have a tool called drawgram for construction of rooted trees. If you want to draw this unrooted tree, okay, you have a program called draw tree. So drawgram is used for construction of phylogenetic trees from Phylip, and draw tree is used for construction of unrooted trees from Phylip. Likewise, we have programs for construction of tree using UPGMA and neighbor joining. For both UPGMA and neighbor joining in Phylip package, we have a program called neighbor. We have a program called neighbor, which is used for construction of trees using both neighbor joining and UPGMA. And we have something called PAUP. That is, if you want to construct phylogenetic trees using parsimony method, you can use this. And if you want to use likelihood method, you can use PAML. So maximum likelihood can be done using PAML. So likewise, we have methods for each and every methods. We have a different programs. Okay, and for calculating protein distance, you have protist, and for calculating DNA distance, you have DNA dist. Okay, and so likewise, we have 35 programs in filing.
Sir, excuse
So,
me.
yes, sir. Yes, madam.
PAUP, is there any example for PAUP?
No, this PAUP and PAML is not from Phylip, but these are exclusive tools. Phylogenetic analysis using parsimony. This is a exclusive tool, not from the Phylip, but even in Phylip, for parsimony, we have a tool called ProtPass. ProtPass, that is protein parsimony. If you're using a DNA sequence and want to go for parsimony method, one can go with DNA parts. So these programs are present in Phylip package, but PAUP, PAML are exclusive softwares. So in internet, if you type PAUP, it gives you a different URL. PAML a different URL. You can try with this also. But in common Phylip URL, one can try with draw program for rooted, unrooted draw tree, protein distance, DNA distance. You use these programs, and for parsimony, you use this, and for maximum likelihood, you use protml and DNAML. These are the programs you can find in the Phylip package. In a single package, you find multiple programs, okay, to work with each and every method. Even we have Fitch-Markolish. To work on Fitch-Markolish, one can use Fitch program, okay, present in Phylip. So for each and every method, you have a program in Phylip. Okay. In Phylip, mostly we try to use download and use the standalone software for practicing. So one can find easier, okay, with Mega Tool, because in Mega Tool, one can able to find, you can work on the types of alignment like cluster W, muzzle, and even you have plenty of options to visualize the phylogenetic trees. Okay. So I have completed my today's session on these methods. We have some description on uh, the character-based methods called parsimony, likelihood, etc. So this we'll be discussing it on tomorrow. So tomorrow we'll be discussing on the character-based methods, and we also see how to solve problems using UPGMA method. Okay. Any doubts, you can ask me at present. Do you have any doubts? If no doubts, thank you very much for your patience even during the holiday. Have a great day to you all. Bye.